Microorganisms. Introduction. Doctors often advise us to boil water before using it for cooking or drinking purposes. The reason is that most of us think that tap water is pure simply because it appears transparent. But in reality, water could contain numerous disease-causing germs. Boiling water helps to kill these germs. These germs are so tiny that they are not visible to the naked eye. We need a microscope to view them. Organisms that are visible only through a microscope are called microorganisms. Microorganisms are also useful to us in many ways. While some help in carrying out biological processes, others help in the preparation of important food items such as curd and bread. Microorganisms are classified into 5 major groups. They are bacteria, fungi, algae, protozoa, and viruses. Forms of bacteria. There are 4 forms of bacteria. Coccus. These bacteria are spherical, round in shape. The spherical forms occur either singly, called micrococcus, in groups of 2, called streptococcus, or in groups resembling bunches of grapes, called staphylococcus. These are shown. Bacillus. These are rod-like, with or without flagella. They may occur singly or form a filament. Certain notorious diseases in man, such as tuberculosis, tetanus, diphtheria, leprosy, are caused by Bacillus bacteria. Spirillum. These are twisted like a corkscrew or spiral in appearance. They may have flagella at both the ends. Vibrio. These are comma-shaped bacteria. Curd formation. Curd is formed when a protein called casein present in the milk clumps together or coagulates to form a solidified mass. The spoonful of curd that your mother adds to the milk activates the growth of lactobacillus, an acid-producing bacterium in the mixture. When casein clumps together, it traps fat globules and some of the milky liquid inside the clumps. This is what gives rise to moist, nutritious curd. Milk plus a little curd, starter, gives to coagulation of milk casein, gives to more curd. Fermentation. Fermentation may be defined as the process of decomposition of sugar molecules by microorganisms to produce an acid or an alcohol. Sugar plus microbe gives to acid or alcohol plus carbon dioxide. Curd and cheese in industry. Curdling of milk is an important process in the preparation of a variety of dairy products such as cheese and paneer. To increase productivity, curd and cheese manufacturers add a starter culture of bacteria, lactobacilli or streptococci, and a substance called rennet obtained from the stomach lining of young cattle to induce lactic acid formation. Curd and yogurt. What we in India called dahi or curd is referred to as plain yogurt in many countries. Like curd, yogurt is also prepared from the bacterial fermentation of milk. Yogurt is often sold flavored with fruits, vanilla, or chocolate, although unflavored yogurt is also quite popular in many countries. Yeast in food industry. Bread production. Kneading a mixture of flour, salt, sugar, yeast cells, and water into dough is an important step in bread production. The process responsible for the fluffiness of bread is the decomposition of sugar by yeast cells to produce carbon dioxide gas and alcohol. When the kneaded dough is shaped and kept in a warm, moist environment, the yeast ferments the sugar to produce alcohol and bubbles of carbon dioxide. Baking the expanded dough at 1800 degrees centigrade kills the yeast and stops fermentation. The alcohol evaporates to form special compounds which impart taste to the bread. Other uses of yeast. Since dough making is an initial step to the preparation of cakes, donuts and buns, yeast cells are considered an important ingredient in the baking industry. In wine-making industries called breweries, sugar present in barley and grape is fermented by yeast to give alcohol and carbon dioxide. Some Indian foodstuffs like idli, dosa, popular in South India, and dokla, popular in Gujarat, make use of the process of fermentation for their preparation. Fermentation of sugar. Aim: to observe increase in volume during fermentation of sugar by yeast cells. Materials required: 1 cup of sugar, a glass of warm water, and active yeast cells. Procedure: add 3 to 4 spoons of water to 1 cup of sugar. Add yeast cells to this mixture. Mix the ingredients thoroughly. What do you observe? Why is there an increase in the volume of the mixture? This happens because active yeast cells, when mixed with sugar and a warm liquid, are given all the basic life support they need: food, moisture, and warmth. The yeast cells immediately feed on the sugar in the mixture and in the process give off carbon dioxide and alcohol. As the gas rises up, we observe an increase in the volume of the mixture. Microbes and its uses. Bacteria is used in the production of vinegar, coffee, and tobacco. Xanthium gum, obtained from the bacteria Xanthomonas campestris, is used as a stabilizing agent in toothpaste. It is also used as a food additive in jams because of its ability to thicken liquid solutions. Fungi. In addition to aiding fermentation, yeast also imparts flavor to wine, beer, cheese, and soy sauce. Some varieties of mushroom are eaten as food. Yeast is used in the production of riboflavin, a type of vitamin B1. The fungus Penicillium notatum, issued in the manufacture of penicillin, the first antibiotic to be discovered. Budding of yeast cells. Put some baker's yeast in a warm sugar solution. Leave it undisturbed for 2 to 3 hours. Put 1 drop of it on a clean glass slide and examine the fluid under a microscope. What do you observe? You will see the budding of yeast cells. Algae. Algae is an excellent setting agent of jelly obtained from the cell walls of a species of red algae. It is used as a thickener in soups, jellies, ice cream, and fruit jams. Brown algae are a rich source of iodine and potassium. Protozoa. Protozoa are simple, minute, unicellular, microscopic animals. They are solitary or colonial, free-living, parasitic or symbiotic. The animals can be sedentary or capable of locomotion. They may move with the help of pseudophodia, flagella, cilia, or by the contractibility of the cell itself. They do not possess a cell wall. The figure shows some forms of protozoa. Euglena is a unicellular organism which may be regarded both as animal as well as plant. This animal possesses an oral opening and a chloroplast. That is why it is known as a biological puzzle. Several protozoa cause diseases in humans and in other animals. Microorganisms. Scrape some slime from the surface of a leaf or stone in a pond with the help of a knife. Place it on a slide with a drop of water. Observe it under a microscope. What do you see? You will see small organisms moving. Try to locate an amoeba and observe its behavior. Viruses. Viruses are found in plants, animals, and in human beings. They are also found in bacterial cells, where they are called bacteriophages. Shape. They have a variety of shapes. They may be rod-shaped, polygonal, spherical, or cubical. Some forms of viruses are shown in the figure. Structure. Their structure is very simple. There is just a core of nucleic acid, RNA, ribonucleic acid, or DNA, deoxyribonucleic acid, surrounded by a sheath of protein. There is no cytoplasm, nucleus, or cell membrane. Size. Viruses are extremely small, smaller than the smallest bacteria, and can be seen only under electron microscope. Their sizes range from 0.015 to 0.2 microns. Harmful microorganisms. Have you ever opened a packet of bread only to find it spoiled and covered with a greenish-white spongy layer? The spongy layer is a form of fungus called the bread mold. Not only fungi, a host of other microorganisms tend to grow on food items, resulting in food spoilage. Microbial growth, especially fungal and bacterial growth, can be a big menace, as it not only destroys the food but also causes many foodborne diseases. A moderately warm temperature, coupled with air and a moist place, is all that most microbes require to thrive and reproduce. Microbial impacts. Besides spoiling food and causing foodborne diseases in animals, microbes can cause diseases in plants too. One such disease is the late blight of potato caused by the fungus Phytophthora infectans. Detection. Food spoilage. A few indicators that tell us whether or not a given sample of food is suitable for consumption or not are listed in the table. They are odor, putrefaction, sliminess, discoloration, souring, and gas formation. Food preservation. Food preservation is the process of treating and handling food with an aim to stop or slow down its spoilage when maintaining its nutritional value, texture, and flavor. Methods of food preservation are tabulated as shown. They are boiling, dehydration, refrigeration, canning, chemical preservatives, and salting. Expiry dates. We notice that most of the packaged food items that we get from the market have best before or expiry dates on them. Expiry date refers to the date before which the supplier intended the date because they are likely to have deteriorated either in flavor, texture, appearance, or nutrition.